Welcome to our presentation, Ken and Professor Lee. Our presentation is the discoveries of antibiotic-producing bacteria in soil by Zaira Ramirez and Rachel Romero. Introduction. The goal of our Tiny Earth Project discoveries was to take what is in our environment, which in this case is our soil, and use those resources to find potential new antibiotics. This is an essential part of our discovery because our world is always evolving. This also involves our antibiotics. Many antibiotics that once were able to do their function and become resistant. Hence, us trying new ways, methods, and new antibiotics. And I added those little pictures to represent the antibiotics versus, bacteria because they're becoming resistant. So, yes. Soil sample location. For my soil sample, I collected my sample on campus by Pioneer Hall. The weather on this day was sunny but not warm. It was 63 degrees Fahrenheit. For Raisha, she collected her soil from the base of a tree in front of Whitman Hall on campus. The weather was in the 50s and it was a cloudy day. My apologies. Methods. Diluting our soil and making a master plate. We first measured our soil to get 1 gram of soil and then proceeded to add that gram of soil to a conical tube with some water and then mixed it in the vortex. We then used a pipette to take 100 liters of our soil and water mixture and put it into a microcentrifuge tube. We then mixed that and kept repeating those steps twice more to dilute our sample. After that, we then used a petri dish to grow our bacteria. We took 100 liters of our solution and put it on our petri dish. After that was on our petri dish, we then used an L-shaped spreader to spread our solution evenly on our petri dish. We used this method with 3 different plates of 3 different dilution factors: 1 to 1,000, 1 to 10,000, and 1 to 100,000. After a week, the colonies of bacteria grew and we were able to get up a new petri dish in a grid format. Using an inoculating loop, we picked individual colonies from the first plate to make a master plate of around 25 different colonies in a grid, making sure to sterilize the inoculating loop in the incinerator between each passing. So it'd be sterile before we move it over. And then for Master Plate 2 and CFU, we were given a new petri dish to make a new Master Plate 2. To make this Master Plate, we used an inoculating loop to move our colonies over from Master Plate 1 to Master Plate 2 in the same order. We had to make sure to avoid any Bacillus mycoides or any fungi that we may have accidentally picked from our soil dilution plate and not transfer it to our second master plate. To calculate the CFU, count and multiply the number of colonies by the dilution factor of the plate we picked to get the CFU / gram of soil.
Setting up our test plates 1 and 2. We were given 2 new petri dishes and labeled each test plate 1 and 2. We then used the bacteria from our master plate number 2 to transfer over onto our test plates, making sure they were in the same exact order. We used the same methods as before, picking and patching. We did both plates at the same time without repicking in between plates. After 24 hours, 7 milliliters of soft agar inoculated with 150 milliliters of 2 safe relatives were poured over the test plates. Methods. Making of our streak plate. We labeled our new petri dish streak plate and made 4 quadrants to see our different results. For our streak plate, we used the same methods as before with an inoculating loop, but this time we only transferred over the bacteria on our master plate number 2 that we saw had a zone of inhibition. We spread this colony to the first quadrant and spread it only in that quadrant. Then we sterilized our loop and took the bacteria we spread on our first quadrant to spread on our second quadrant. We repeated this step to the remaining 2 quadrants, making sure we sterilized our loop every time. In the top right of the slide, you can see our streak plate method with each color representing a different time we sterilized the loop. Making a streak plate number 2 / metabolism array. We chose an isolated colony from our streak plate number 1 to safely transfer over to our streak plate number 2 using the same method as our first streak plate. We used those same colonies from streak plate 1 to inoculate into 2 separate tubes of agar. One was labeled anaerobic and the other was aerobic. The anaerobic tube got 20 drops of mineral oil so that oxygen could not reach the bacteria. Setting up a PCR and gram staining. To set up the PCR, we took a sample of a colony from our second streak plate using an inoculating loop to mix it with a small amount of water. These were then boiled to separate the DNA.
For my results, I tested a total of 25 bacterial isolates. The escape pathogens I tested, my apologies, were Bacillus cereus and E. coli. I was not able to find any antibiotic producers. I also did not have any zones of inhibitions. They were vaguely shown, but not proficient enough to move on with my study. On that, the bacteria turned out to be gram-negative with the morphology being bacilli, as shown there. Yes, after getting our PCR data, I unfortunately was not able to find any discoveries. This could have been due to the lack of genetic information to produce a solid estimate answer. I'm not really sure. I was a little bummed out that I could not find anything, but that still does not discourage me from maybe trying again in the future. I also found out through my testing that my bacteria is capable of aerobic respiration. I know this is because of the change in the color of my test tube, and hence the color in my test tube of anaerobic did not change. Hence, there was a change in the aerobic telling me there is aerobic respiration.
My results. I tested a total of 25 bacterial isolates. The escape pathogens I tested against were Pseudomonas putida and Staphylococcus epidermidis, and I was able to find one antibiotic producer against Staphylococcus epidermidis. In the picture bottom right, you can see it circled. The bacteria turned out to be gram-negative with the morphology being diplococci. After getting our PCR data, I discovered my bacteria was Variovorax sp strain LY1. Some other information I found out about my soil bacteria was that the CFU was 44,300,000 CFU per grams of soil, and that the bacteria is capable of anaerobic respiration. Discussion. Through this experiment of the Tiny Earth Lab, although Zyra was not able to find her bacteria due to insufficient evidence, we're still very glad she was able to take part in the experiment, and it was a learning experience and one that she enjoyed being a part of. I was able to conclude the type of soil bacteria I found, as well as many aspects of it. I appreciate the amount of information I was able to get from just a sample of soil. For potential further steps, we could test our soil samples against different escape pathogens in order to get different results and see if maybe we just didn't test against the right pathogen. We could also try different soil samples or get soil from a location off campus. In conclusion, although one of us was able to find discoveries and the other was unable, this does not mean there was nothing to find or give up on. We learned a lot through the processes and methods of various experiments we did to identify parts of our soil bacteria. This was an interesting experiment and a learning experience with trying new methods to try out in lab. Thank you for watching.